and we are thrilled that Dr. Florian Krammer is with us again, has broken the record for the person to have spoken at Grand Rounds more times than any other living human being. And we are really thrilled that he is with us. He is a PhD who graduated from the University of Natural Resources and Life Sciences in Vienna in 2010. He received his postdoctoral training in the laboratory of Dr. Peter Police at the Icahn School of Medicine here at Mount Sinai. In 2014, Dr. Krammer became an independent principal investigator and is currently Mount Sinai's professor of vaccinology. Dr. Krammer's work focuses on understanding the mechanisms of interactions between antibodies and the viral surface glycoproteins, and on translating this work into vaccines and therapeutics. His main focus is the influenza virus, but he has also been called upon now to work on coronaviruses and a bunch of other viruses that I can't pronounce: flaviviruses, hantaviruses, filoviruses, and arenaviruses. He's published more than 240 papers on these topics. As principal investigator of the Sinai Emory Multi-Institutional Collaborative Influenza Vaccine Innovation Center, which develops improved seasonal and universal influenza virus vaccines that induce long-lasting protection against drifted, seasonal, zoonotic, and future pandemic influenza viruses. In response to the pandemic, Dr. Krammer's lab worked swiftly to respond, developing the first serological assay to receive FDA emergency use authorization. Dr. Krammer's also been involved with pandemic-related science outreach in the United States, Germany, and Austria, and has participated in congressional hearings and other policy initiatives. We are really thrilled that he's part of our Sinai family, and I turn it over to you. And thank you so much for joining us so early in the morning.
Thank you very much for the introduction. Okay, let me try to share my slides. I think this should work. Okay, can you all see the slides?
Yes.
Perfect. Okay, so what I thought I would do today is basically give an update of all the news that we have about the new variants, new vaccines, what's happening there, and also a little bit what's happening with the evolution of SARS-CoV-2. Before I start, I just want to put up here my disclosures. Mount Sinai has filed patent applications on SARS-CoV-2 serological assays and Mucosal disease virus-based vaccines. That's not going to be the main topic here. I'll talk a little bit about serology, but not necessarily related to our assays. All right. I just wanted to look back and see where we stand with SARS-CoV-2 globally right now. As you all know, this has now been a problem for almost 2.5 years. We are now officially at about 540 million infections worldwide. Of course, there are many more infections. Those are just the official numbers. And globally, we had about 6.3 million deaths officially, and I'll get to the unofficial numbers. In the US, we had more than 1 million COVID-19 deaths, which is really, really high. That's higher than what we had for the 1918 pandemic. If you don't normalize it to population size, it's still below that if you normalize for population size. What you see here is the global epidemic curves, cases per day on the left side and deaths per day on the right side for the last 2.5 years. And what you can see here on the left side is, of course, these different waves that we had. And this last really big peak here is the Omicron BA.1 virus. Excuse me. And the second wave here that you see is the Omicron BA.2 virus. And there's this little wave here on the right side. These are Omicron subvariants. And this is what we see for infection. And of course, there was a huge number of infections with Omicron. And then here on the right side, we have deaths. And of course, there were more deaths before with other variants. And there's actually a proportionally lower impact of Omicron if you look globally. And this might have to do with and we'll get into that with the little bit less severity of Omicron, but it's still very dangerous. And of course, the baseline immunity that we have now in the population. And you see, it's actually really nice to see this. The numbers of deaths per day globally really go down drastically now. These are the official numbers, but there's also estimates based on excess death records. And so there are two studies. One is by The Economist, and another one is a scientific paper in The Lancet. And by the end of last year, they estimated that about 18 million people died of COVID-19 globally. And of course, now this number is even higher. So that's a relatively large number. Again, still not where we were with the 1918 pandemic, where the estimates are somewhere between 40 and 100 million deaths globally, but unfortunately, this is increasing. Of course, a lot of countermeasures have been developed, and the most important ones are vaccines. We have now in the US 3 authorized vaccines. 2 were mRNA vaccines. This percentages here is the initial efficacy from the clinical trials. So the mRNA vaccines worked really, really well. We also had the J&J vaccine in the US, which is a viral vectored vaccine with lower efficacy, but still holds up relatively well. And then there's a lot of other vaccines that are used globally. And in fact, the AstraZeneca vaccine and these inactivated vaccines that we see here that are mostly produced in China, but also in India, are the vaccines that are mostly used globally. So the US is and also Europe is very focused on the mRNA vaccines, but the heavy lifting globally has really been done, was really done by these inactivated vaccines and the vectored vaccines. But again, there's a large diversity of vaccines. There's even more than what I put here that are used worldwide. And we might get another vaccine candidate here in the US on the market soon, and that's the Novavax vaccine, which is a recombinant protein vaccine. This is already used in Europe and in a few other countries, and has worked really well, similar to the mRNA vaccines. Now, at the same time, when we started to use these vaccines, in the end of 2020, the first viral variants emerged, actually a little bit earlier even. And the first variants that we saw were the alpha variant, or P117, the beta variant, or P1351, or the gamma variant, P1. And so we really started to discuss these in detail in December of 2020. I remember it was around Christmas, and all of a sudden we had all these WHO and NIH meetings the whole day to figure out what the impact of these variants could be. And so the alpha variant showed up first in the UK, and the main point here was that it had a mutation in its receptor binding domain. So here on the right side, the structure is the spike protein, which the virus uses to attach to our cells. And this here is the receptor binding domain. So the little piece of the spike that actually interacts with the receptor, with ACE2, right? And so this interaction here is really important. And the alpha variant had a mutation here, basically from N to Y, and that increased the affinity of the receptor binding domain to the receptor. And that probably caused this variant to be more infectious. And so it spread quicker, and that's how it was detected. And then we had the beta and the gamma variants, which had additional mutations, and that meant that they could escape partially from neutralizing antibody responses, not completely, but partially. As you know, these were variants of concern. The WHO determines what a variant of concern is, and then we also have variants of interest. And there were many more variants of interest, including lambda, mu, epsilon, zeta, eta, theta, iota. This was our New York local variant, and kappa. And they caused some issues, but they never really took off. And so in the end, none of this was really a big issue for vaccines. Basically a lot of these variants just completely disappeared. The real trouble started with the rise of the Delta variant. And so you remember last year in spring, we had in the US this little spring wave, and then case numbers were dropping and more people were getting vaccinated. And a lot of people, including me, were thinking, oh, now it's going to be over soon. It's great. But unfortunately, what happened, there was a big epidemic in India at the same time. And there a variant emerged that was called Delta, or B1617.2. And this variant is not so much an escape mutant from immune responses or escape variant from immune responses, but it's very infectious. And it started to spread globally. And as you remember, in the beginning of the summer of 2021, we started to detect this variant in the US, initially at a low frequency here in the beginning of June. It's about 10%, but then this expanded very quickly, right? And then within a few months, it was the dominant variant. And again, this wasn't necessarily escaping from immune responses so much, but it produced very high titers in the upper respiratory tract. And that's actually also really when we started to see the first breakthrough infections. Again, the vaccine was still working well against it, but of course, because it was so infectious, it caused a relatively big wave, especially in the south of the US in summer. In New York, it was less of a problem. And then in November of 2021, Omicron happened, and Omicron was really very unexpected. So on November 22nd, and I remember this well, I was on Twitter, and there's this guy from the UK, Tom Peacock, who's an evolutionary biologist. And so he pointed out there was a new sequence in an international database, only a partial sequence from a traveler who traveled from South Africa to Hong Kong. And this had an enormous number of mutations in the spike. And I looked at that, and I'm part of a group that actually tracks variants for the US government. And I shared that with the group. And then just on the next day, Botswana and South Africa, specifically South Africa's really good genomic surveillance, better than the US, they found a lot of these sequences. And they had a wave that was ongoing. And they concluded that their ongoing wave is really based on this new variant. And you can see here, I won't go through that, but there was a huge number of mutations there, and the virus variant was called B11529. On November 25th, so that was Thanksgiving, there was a press conference in South Africa where they basically said, okay, we have this new variant and it's causing a big wave. And at the same time, Israel and Belgium already found cases of this variant. So meaning that it had already spread internationally. And on November 26th, the WHO met and announced that this B11529 is going to be called Omicron, and it's going to be a variant of concern. And the rest is really history. This virus spread very quickly. In the middle of December, we already had a big Omicron wave starting here in New York. Why are we worried about this virus? What happened really? On the right side, you can see the structure of the spike protein of Delta, and in red you can see the mutations that it has compared to the original virus, right? And so we have these two mutations in the receptor binding domain, which are not really a big deal, and then there's some mutations outside of the receptor binding domain. If you compare that to Omicron, Omicron is really a monster. There's 15 mutations in the receptor binding domain. There's a large number of mutations outside of the receptor binding domain. And when we looked at the sequence, if you're familiar of where on the spike these neutralizing antibodies bind, you quickly realize that all of these sites where neutralizing antibodies bind have been changed. And that was, of course, a huge concern that this virus might cause a large number of breakthrough infections just because it escapes immune responses very, very well, and it's still very infectious. And so what were the consequences? Typically double vaccinated individuals had little protection from infection. And I'm saying infection here. Disease or severe disease is a very different story, and we'll get to that. But there was very little protection from infection anymore. There were a lot of breakthrough infections, and I mean, all of you have experienced that. Many of you might have had breakthrough infections. Omicron and its subvariants have no problem infecting somebody who has had 2 shots of mRNA vaccine. The booster dose, a third dose, actually increases protection from infection again. And I'll show that in a graph soon. But this protection wanes quickly. Reinfections were relatively uncommon before Omicron. You had good protection from reinfection, even with the Delta variant. But for Omicron, these are now relatively common. So it's the same problem as with vaccines. On the plus side, having baseline immunity, either through infection or vaccination, seems to protect well from severe disease. And that's true for all Omicron variants. If people haven't been infected before, if people haven't had been vaccinated, and now they get infected with an Omicron variant, they mount very Omicron-specific immunity, and it's very likely that that they can get infected with future variants again. So that's also a thing that we learned. And in general, Omicron causes less often severe diseases compared to Delta. Now, I think there was a little misconception about that in the beginning in the media. So there is indeed a reduction in severe cases of about 70% if you compare Omicron and Delta. But Delta was more pathogenic than, let's say, Alpha. And Omicron is basically back at the level where Alpha was, right? So it doesn't really mean that it's a harmless virus. It's just the frequency is a little bit different. I'll show you an example of what happened in countries where you can really compare that. But what we have to keep in mind is that the vaccines still protect, not from infection necessarily, but from severe disease. And so what I'm showing you here in the upper panel are data from the UK of vaccine effectiveness against Delta in black and Omicron in gray. And you can see on the left side what happens after 2 doses of vaccine. Of course, if you get 2 doses of vaccine, there's pretty good protection even after a long time against Delta. But the protection against Omicron wanes very quickly. So basically goes to 0 over time. Now, if you get a booster dose, of course, you have very nice protection against Delta again. And initially, you have very nice protection against Omicron as well, but this starts to wane. More so, it looks for a Pfizer booster than for a Moderna booster, but in both cases, it goes down. But the important point is here, if you have 3 shots of mRNA vaccine, the protection from mortality is still very, very high. And so this is data from the UK. They do a very good job of monitoring that, but so does New York State. A little bit different. They don't differentiate between 2 shots and 3 shots and what type of vaccine, but they actually measure vaccine effectiveness per week. Every week. You can look that up on their homepage. The link is here. And so what they saw initially was when they looked at vaccine effectiveness against basically infection and disease, you see in the beginning this was really high. So this is spring of 2021, we're at 90%. And then over time this starts to fall. It goes down to approximately 80% with Delta. And then it further goes down with Omicron. And so this is basically the red bar here indicates the Omicron era, and you can see it falls from about 80 to about 70%. The important point is here on the right. So this is vaccine effectiveness against hospitalization, and we start very high. We start above 90%. And we end here with about 90%. And that's important too, right? So there's much less of a drop in vaccine effectiveness against severe disease. Now, a very important example here. There are a number of countries that had a zero COVID strategy, right? Those were usually small countries, often islands that had an easy time to close their borders. But 2 of them, Hong Kong and New Zealand, really did a great job of keeping the virus out for a very long time. And that didn't work anymore with Omicron because the virus was very infectious, right? And so at the same time, both Hong Kong and New Zealand had a wave of Omicron. You can see that here on the left side, the curve shapes are a little bit different. For Hong Kong, it's a steep increase and then it falls very quickly again. For New Zealand, it's a little bit more gradual, but they both, and this is normalized per million inhabitants, but they both had huge Omicron waves. This is cases on the left side, and here on the right side you have deaths. And there's this huge difference, right? There's a lot of deaths in Hong Kong. There's very few deaths in New Zealand. Now, what was the difference there? At the beginning of this wave, Hong Kong had vaccinated 30% of the 80 plus population. New Zealand had literally vaccinated 100% of the 80 plus population. And that really made the difference. It didn't protect these people from getting infected, but it did protect these people from dying. And this is, I think, even now that the virus changed so much, we still see that the vaccines have a very good protective effect. Now, I want to talk a little bit about Omicron and its subvariants, right? You see this phylogenetic tree here. We have Delta here on the left. We have a few other variants here, and Omicron is here on the right. And what you can see right away is that there is two subvariants that showed up initially. The first one was BA1, which caused the first wave, and Omicron BA2, which was causing the second wave. And in addition to that, we had subvariants of those two. There was a BA1.1, which had an additional mutation that's now gone. And what is really prevalent in New York, and this was kind of a local variant, was BA2.12.1, and that had additional mutations as well. This is still circulating in New York. And then in addition, we have BA5, BA4, and BA5, which are now coming up here. This is a similar tree, just showing you the relationship here again. Here you have BA1, here is BA2, and then BA4 and 5 are related to BA2, and then BA2.12.1 is of course also related to BA2. There was a BA3, but that never took off. And this is, I got this from Harm van Bakel and Viviana Simon, who are involved with the Mount Sinai pathogen surveillance program. This is the prevalence here right now in New York of these different variants. So this is not quantitative data in a way that it doesn't tell you how much virus is circulating. It just tells you the proportion of the different variants of the virus that is circulating in a given week. And you can see here we have early in the year, this BA1 dominance, then BA2 came up, screen, this green color here. Then we had our own local variant taking over BA2.12.1 here in teal. And then now we slowly start to see BA4 and BA5 creeping up. And we have also detected Omicron XE, and we'll get into that. That's a recombinant virus. What do we need to keep in mind when we think about these different subvariants? So the issue that we see, and the focus on the upper panel here, is that first of all, a lot of the monoclonal antibodies that have been used as therapeutics started to not work against Omicron anymore, right? There was one exception that was initially still used, and that's Sotrovimab, or here it's called S309. That's the name that we use for it in the scientific literature. And you see this worked well here against wherever it says T614G. This is wild type, or the original virus. And basically, the higher the points here are, the better it neutralizes. And then you can see it drops against the original Omicron, and then it drops further against these subvariants. And that happened with a lot of antibodies that were used. And right now we only have basically one antibody that is useful against all these variants, and that's Bebtelovimab, or as we call it, Icahn 1404. And this is this guy here, these triangles, right? So this neutralizes the original virus very well. It neutralizes Omicron BA1 very well, and it neutralizes basically nearly all the subvariants, including BA4 and 5 very well. So this is still a good antibody to use. And then the other interesting part is Evusheld, which is actually a mix of two different antibodies. Evusheld is given prophylactically, and the antibodies that are used in Evusheld, that is COV2130. Let me just look for it really quickly here. 2130 is here. So this antibody didn't work well against BA1 and BA1.1, but it still works against the new subvariants, so we're lucky. And then 2196 is the second antibody, which is over here, and that doesn't work against BA4 and BA5 anymore, unfortunately. But because one of the antibodies still works well, it is reasonable to assume that Evusheld will still work, although not as good as against other variants. We also now have a lot of data on how well the sera from vaccinated, infected, and so on and so forth, people still neutralize these different subvariants. This is from a paper that David Ho recently published from Columbia. And so what you can see here is if people got 3 shots of mRNA vaccine, of course, they have high neutralizing titers against the original virus. This drops off against the early Omicron variants quite a bit, also against BA2, and it further drops against BA4, BA5, because these viruses have additional mutations, right? People with hybrid immunity, meaning that they had an infection and then a vaccination, have a more equal response. There's also a drop, but there's not much of a drop from, let's say, BA2 to BA4, 5 anymore. And people with breakthrough infections against BA1 still have a sharp drop to BA4, BA5. So these people might actually at some point get reinfected with BA4, BA5. If you had a BA2 breakthrough infection, it looks a little bit better. So, we need to keep monitoring these neutralizing activities of the monoclonal antibodies and also of sera against these emerging variants. There's some more clinical data and vaccine effectiveness data for BA2. We don't have that for BA4, 5 yet, but in the UK, there was an analysis done that compared the risk of hospitalization between BA1 and then BA2 when it showed up, and they didn't find a difference. So basically, BA2 and likely its subvariants behave very similar to the original Omicron. And the same is true in terms of vaccine effectiveness. This is shown here. This compares, again, in the UK, vaccine effectiveness against BA1 and BA2 after the first 2 doses of mRNA vaccines. So it's very similar. It drops sharply. And then again, after the booster dose, where it goes up again, but then also starts to drop again. But there's little difference between the subvariants here. Another interesting thing that you might have heard of and that you might come across is recombinant viruses. And I already discussed one of them, XE, which we are now detecting in New York. And so coronaviruses can recombine genomes, right, if they're closely related. And of course, this works with variants. We had some evidence for this happening early in the pandemic, but now we see recombinants between different Omicron versions and Delta and Omicron versions. And there's 3 that have been officially recognized. One is called the XD, where we have basically the genome or most of the genome from Delta, and then the spike protein basically from Omicron BA1. There's XE, which is around in New York, which is a mix of BA1 and BA2. And then there's XF, which is basically contains a small part of the genome of Delta and then a larger part of the genome of Omicron BA1. And so why is that interesting? As I said, there are differences in pathogenicity between Delta and Omicron. This is very obvious in animal models. So here on the right side, you have data from a mouse model. And this is a Kaplan-Meier survival curve. So when you infect these mice, which are special genetically modified mice, if you infect them with Delta, the mice die. If you infect them with Omicron, the mice survive. They get sick, but they survive. If you use this recombinant XT virus, which has the spike protein of BA1, but the remaining genome part from Delta, the mice die as well. And so this suggests that it's not the spike that makes Omicron less pathogenic. It must be other mutations as well. This recombinant we haven't seen so far in New York, and it doesn't seem like it's increasing in prevalence. I talked a lot about these variants, and I want to go a little bit into where they come from, right? Coronaviruses are actually exceptional among RNA viruses, because most RNA viruses mutate a lot because they don't have a proofreading activity. So they have a high error rate, and these errors, when they copy their genome, are not corrected. Now, coronaviruses are an exception because they actually do have proofreading activity, right? Errors are typically corrected, and so they mutate slower. And so that's why a lot of people were surprised to see all these variants in the beginning, in the end of 2020, because the assumption was the virus is not going to mutate that much. But there are two factors that drive that. One is if you have explosive spread, like in a pandemic, when you have a lot of cases, you have a lot of infections, and every infection involves many replication cycles. And every replication cycle, every time the genome is copied, is a chance for the virus to make a mistake and introduce a mutation. But there's an additional point here, and that is that we see persistent infection in immunocompromised individuals, and it seems that fast evolution happens there. And here on the right side is just an example. This is a New England Journal of Medicine paper from 2020, where an immunocompromised patient was infected for more than 150 days. And when virus was sampled different days, you can actually see that these mutations that we also see in variants at position 484, for example, or at position 501, these mutations come up in these persistently infected individuals. And so for a very long time now, the hypothesis was that these variants really come out of immunocompromised, persistently infected individuals that get infected. They don't die. They might actually be relatively okay. They mount some type of immune response against the virus, and that puts evolutionary pressure. And then the virus over time adapts and adapts and adapts, and at some point it jumps to another person with all these mutations, and then it can start spreading. And that's the idea how these variants are generated. And I wanted to show you an example. This is actually very exciting work by my colleagues Viviana Simon and Harm van Bakel and Mia Sordillo. And so they detected also through the Mount Sinai pathogen surveillance program, they found a patient who was infected in December with Omicron, and then was infected until March persistently. And they found that this is Omicron BA1, they found that over time, the virus that was circulating in this person was accumulating 8 additional mutations in the spike protein. So basically, they caught the variant in the making. And the scary thing about this is this virus actually transmitted. So they found this virus also in 4 additional individuals. And then the CDC found it in additional samples from the population in New York, right? So basically telling you that this virus was fit and was spreading. We haven't seen it now for a few weeks, but this doesn't mean much because, of course, we sequence only a very small proportion of infections. And here's a phylogenetic tree of that. So the red line is still one and the same patient. So you see evolution happening there. And then you see here the tree branches for these other people who got infected. Person 1, 2. Person 1 is still the original person 2, person 3, person 4, and then these 2 here are the sequences from the population that the CDC detected and deposited in a public database. So the take-home message here is, if you have patients who test positive for many months and who seem to be okay, they might be a threat for global public health, because they might actually breed a variant, and that variant might transmit on. We don't have a good idea of how to get rid of these persistent infections yet. There might be a chance to get rid of that with Paxlovid in combination with monoclonal antibodies, but don't just assume that these people are fine because they seem to be healthy. If they test positive for many months, there's something wrong, and we need to try to get rid of that virus in this person. And that brings us a little bit to evolution and to trying to figure out what is going to happen in the future. So for a lot of viruses, we see this ladder-like evolution, right? So you have one virus, and the virus mutates, and it generates a variant that is basically a descendant of this original virus. And then there's another set of mutations, and another set of mutations, and another set of mutations, and you get this ladder-like evolution. And this is relatively easy to predict, and it's very easy to update vaccines for something like that. Other viruses, influenza B, for example, did that, split up in lineages, but then behave the same. They have this ladder-like evolution. And again, this is more complicated, but it's still easy to predict, and you can still update vaccines nicely. But then we have cases in influenza H3 and 2 did that before the pandemic, where we have a very chaotic evolution, where there's many different variants that co-circulate, and you don't know what will become dominant. And this is, of course, a very difficult situation when you try to predict what's going to happen when you try to update vaccines. And unfortunately, that's exactly what we see with SARS-CoV-2. We have this almost star-like evolution, where here in the middle is the original virus, and then all the variants so far just came out from the middle, right? They're all related to the original virus, but they're not necessarily related to each other, right? The Omicron variants are not descendants of Delta. They are descendants of the original virus. Delta is not a descendant of Alpha or Beta or Gamma. It's coming from the original virus. And that makes predictions really hard, right? Because if you would think that, and we don't know, I mean, we now see subvariants of Omicron, and they might dominate in the future, but we have seen the same before with Delta, and we assumed everything that's now coming is going to be descendant of Delta, and that wasn't true. And the problem really here is these persistently infected individuals that might still carry the original virus from 2020, right? And so this makes it very unpredictable, and that's, of course, highly problematic. But to have a more general look into the future and what might happen, we can look at other seasonal viruses, respiratory viruses, and specifically at human coronaviruses, right? There's 4 of them. You might be familiar with them: 229E, HKU1, NL63, and OC43. 2 of them, OC43 and HKU1, are relatively closely related to SARS-CoV-2. And they all are seasonal, right? Their season starts in fall. They peak around January, February, and then case numbers go down. And in summer, we have barely any cases with these viruses. And it's very likely that over time, exactly that will happen with SARS-CoV-2. And we see that to a certain degree already. These human coronaviruses, by the way, can cause up to 30% of all common colds. Not in every season, but it can be up to 30%. And another fun fact here is that this OC43 here is a suspect for causing the 1889-1890 Russian flu pandemic. We don't know for sure, but this one is closely related to bovine coronavirus that can infect humans occasionally. And if you look at the phylogenetic, if the sequences of OC43 of this human coronavirus and bovine coronavirus, and you do a phylogenetic clock analysis, so basically trying to infer when they split, you end up with about the year 1880, which would fit well to this. And so this might be an example where this virus originally jumped from cows into humans, caused a pandemic, and then over time became a seasonal coronavirus. We actually think that all 4 of them originally came from animals and jumped into humans at some point in time. So the assumption is this will become a seasonal virus, right? Seasonal respiratory disease. But we have a lot of them, right? And on the one side, we have, for example, these human coronaviruses, but other seasonal respiratory viruses as well that have seasonal circulation that cause relatively mild disease, and you don't need vaccination against them because the disease is in general mild. There might be differences when it comes to immunocompromised individuals, but in general, it's mild, right? And on the other side of the spectrum, we have influenza, which also is a seasonal virus, but it causes substantial severe disease, and annual vaccination is necessary. And so the question really is, where is SARS-CoV-2 going to fall, right? Is it going to be closer to human coronaviruses and will do little damage, although it might cause seasonal infections every year, or is it going to be closer to influenza, where we really have a lot of severe disease and also, unfortunately, deaths? So that's the big question, and we just don't know yet. And so I want to switch gears a little bit from the virus to what we're doing now to tackle these variants and to update vaccines and so on and so forth. So I'll walk you through a few updates. The first one is something that is discussed quite a bit right now, specifically because there was a press release by Moderna this week, and that's the question of when do we get variant-specific vaccines. So the problem is with the vaccines that we currently have, they contain the sequence of the original virus, but unfortunately, the circulating virus is very different now. Omicron has a lot of mutations, and so it's not very helpful to vaccinate with this old virus anymore, with this old sequence anymore, and it would be much better to vaccinate with something that's updated, right? And so companies have started to work on these Omicron-specific boosters or vaccines very quickly after Omicron emerged. Moderna has a bivalent vaccine that they're testing and a monovalent Omicron vaccine. Pfizer also has vaccine candidates in clinical trials. And the problem was that they couldn't just switch the sequence, right? This would be technically possible, but that's not what the FDA wants. They wanted to see clinical trials, and so this took a long time. And this week, Moderna announced that they have positive results for the bivalent vaccine, which is called mRNA 1273-214. And the Pfizer data is likely becoming available by summer, so basically should come out soon as well. The problem was really that, there was no pathway, no regulatory pathway for quick licensure of these updated vaccines. And so the FDA is still thinking about how to do that. And there will be a VRBPAC meeting on June 28th, where they will try to figure out how this data now looks from Moderna and how to move forward with getting these updated vaccines to the market. But it's very likely that in fall we'll have a Omicron-specific vaccine, at least from Moderna. So with their press release, they also released some data, not much, but some. And this is what you can see here. So these are Omicron-specific neutralization titers. And so what they did here is they basically gave a fourth, I believe this is a fourth dose, either of the original vaccine, and you see this boosts antibody titers against Omicron. And then they also gave this bivalent vaccine that has Omicron and the original sequence. And you see that these titers are higher. It's not really hugely higher, but it's statistically significant. You can see that in seronegative individuals, and you can also see that in seropositive individuals. And it's very likely that the FDA agrees with that data and allows them to switch to this bivalent vaccine. So hopefully this will come in fall, and that might protect now better again against mild disease and infection with Omicron. The other thing that is really discussed a lot are vaccines that induce mucosal immunity, right? So when you get a natural infection, the virus enters through your upper respiratory tract, and then you get basically systemic immunity, and that's mostly IgG in serum that also protects the lung very well. But because it came through the respiratory tract and stimulated immunity there, you also get mucosal immunity in the upper respiratory tract, which is mainly secretory IgA, but also T cells. Now, you can imagine if you have this mucosal immunity in the upper respiratory tract that protects you well from reinfection, right? Now, when we get vaccinated with intramuscular vaccines, and all SARS-CoV-2 vaccines right now are intramuscular, we get really strong and robust systemic immunity, right? So the lung is well protected, but very little of that IgG actually ends up in the upper respiratory tract. Initially, when you have really high titers, there might be enough to protect you from infection, but then these titers decline, and there might just not be enough IgG anymore in the upper respiratory tract to protect you from infection. Of course, you're protected from severe disease. Now, a way to fix this situation would be intranasal vaccination, where you mimic a natural infection and you stimulate these mucosal immune responses on the upper respiratory tract. And this is what a lot of companies are now trying to do. So this is a list of clinical trials with vaccines that are either given intranasally or orally or sublingually. And there are a few that are relatively far in phase 3 trials. The problem is that these phase 3 trials are mostly taking place in countries outside of Europe and the US. So this phase 3 trial, for example, takes place in China. Here we have one in India. Here we have one in Iran. And even if they have good results in the end, it's very unlikely that these vaccines will be licensed in the US very quickly. But there's also some developments, including a vaccine that we are developing right now at Mount Sinai, that are ongoing in the US. It might just take a longer period of time until these vaccines are available. So I wouldn't expect vaccines that give you mucosal immunity quickly. This might take 1 to 2 years until the development is ready. There's also a few updates about vaccines for kids. You're probably very familiar with what is used right now. We have the Pfizer-BioNTech vaccine that can be used in the age group 5 to 12 with a reduced dose of RNA, 10 micrograms, which is a third of the adult dose. That works. However, of course, against Omicron, it also doesn't work very well anymore. And the booster dose is now recommended for that age group. Moderna also had clinical trials that they were doing with a 25 microgram RNA dose, and they looked at the age group 6 months to 6 years. They saw relatively low efficacy, but again, this was against Omicron, so this was expected. And they recently submitted their data package to the FDA in April. And there's a VRBPAC meeting scheduled for, this should be June 14 and 15, where this will be discussed. And it's very likely that that vaccine gets licensed. And then for Pfizer, they also tested a 3 microgram dose, so a 10th of the adult dose in the age group 6 months to 5 years. In the very young ones, they had good immunity. In the 2-year to 5-year-olds, the immunity wasn't as great. And so they had to test the third dose in that clinical trial. But there's also, they have been submitting data to the FDA as well. And again, there should be a decision on that on June 14, 15. This should be June and not April. There's also a big discussion going on about the 4th dose. And this, what I show here on the right side, is data from Israel. So now we're talking about the 4th dose with the same vaccine that we're already using. And so Israel rolled that out in older individuals, I think above 60 or above 65. And the data is shown here. What you see here is basically risk reduction as compared to 3 doses, okay? So on top of the 3 doses. And what they show here is that indeed you get further risk reduction when it comes to severe illness. This probably has to do with a lot of people in this cohort that had not responded well to the first 2 or 3 doses, and then the 4th dose, of course, helps. And those are the people who had high risk, a lot of immunocompromised patients, for example. The problem is that overall protection from infection or mild disease is increased, but very transiently. You see that here there's a 2-fold increase compared to 3 vaccinations. around week 3 and week 4, but it's very transiently, and basically 2 months after vaccination, this is going back to what you had after 3 vaccinations. So this 4th dose right now might be useful for elderly individuals or immunocompromised individuals, but it's probably not going to help the general population to get protected against infection and mild disease. I think for that we really need an Omicron specific vaccine. And I'm going back to this. I think I showed this slide when we talked last time. We know that not everybody mounts good immune responses. This is an example from a multiple myeloma patient who had CAR T-cell therapy and then got vaccinated twice, and then unfortunately got infected with SARS-CoV-2 and died. And it turned out this person had no antibodies and no T cells, no antibody response and no T cell response to the vaccine. And a proportion of multiple myeloma patients actually doesn't respond to the vaccine. Since then, we have found that there might be some solid cancer patients, depending on their treatment, that don't respond. There are people who have multiple sclerosis who are on certain treatments, CD20 antibody treatment, for example, who don't respond. And it's really important to figure out if they responded or if they didn't respond. If they didn't respond, they're of course at high risk, and they should know that. And a way to figure that out is basically by running serological tests. And so that's a segue into the last topic that I wanted to cover briefly, and that's correlates of protection, right? And before we talk about that, we need to talk about protection from what type of disease, or from protection from what, right? And so we have basically 3 scenarios. One is protection from infection, and mechanistically you need neutralizing antibodies for that. No other immune mechanism can protect you from infection. Even if infected cells are cleared quickly by T cells, you still get infected, right? So you need neutralizing antibodies for this, and especially on the mucosal surfaces. Then we have protection from disease, and this depends on incubation time a little bit, and I'll follow up on that. But basically, for protection from disease, you probably also need neutralizing antibodies, and plus an anamnestic B and T cell response, and non-neutralizing antibodies can help here too. So for this anamnestic B and T cell response, you need time, right? So B cells, T cells need a few days to really ramp up. Unfortunately, with Omicron, the incubation time is now short. It's only 3 days anymore, sometimes even 2 days. And that's not enough time for B and T cells to really ramp up and respond. And that's probably why we see much more breakthrough in terms of mild disease, in addition to the immune escape properties of Omicron. And then for severe disease, the situation is very different. Here, very likely an anamnestic B and T cell response and non-neutralizing antibodies that can bind broader are the mechanisms that protect you from progressing to severe disease. Just to go back to the neutralizing antibody response, from a scientific point of view, it's very clear that neutralizing antibodies are a correlate of protection. This has been very well established for the original SARS-CoV-2 variant. Here on the left side, you have one graph where on the x-axis you have neutralizing activity against the virus, and on the y-axis you have basically risk of getting infected. And you can see this line, the further it goes to the right, the more it drops. And above 1,000 neutralizing units here, there's basically detected no infections anymore. And you can also see that from other studies where there's a very nice correlation between protection and either neutralizing or even binding antibodies. And so this has been scientifically established, but there are a lot of hurdles to actually put that in use clinically. One is that targets of serological assays often are not unified. Some people still measure antibodies to nucleoprotein. Some people some tests measure antibodies to receptor binding domain, some to spike. Not all serological assays are quantitative, and the implementation of standards so that you have the same units for all tests is not widespread in the US. Europe is a little bit better with that. And as I will show you, the variants make everything more complicated. Even if you look at it from a scientific point of view. And again, going back to what are the endpoints, right? Are we talking about protection from infection, protection from disease, or from severe disease? And so in the beginning of the pandemic, when you measured binding antibody, you basically indirectly also measured neutralization. This is shown here. We have a binding antibody here on the y-axis, neutralization on the x-axis. This is data from our pathology department. And, you know, in the beginning, there was a really nice correlation, right? So you knew if you had higher binding antibody, and that's what you get when you send serum to laboratory, you also had higher neutralization. Now, the variants changed that a little bit. First of all, there's a right shift in vaccine effectiveness, right? So there was this nice correlation here between neutralization and efficacy for the original virus, and then basically the more these variants differ and the more they escape, the more of a right shift you have, and this relationship between binding and neutralizing antibody also changes, right? So initially, this was a very good correlation. And then here, what you can see is that neutralization against Omicron dropped quite drastically, right? Often more than 20-fold. But binding activity against Omicron dropped very little, right? 2.5-fold. So there's a 10-fold difference here. It's a factor 10. And what that does is this the slope of the correlation is still there, but the slope of the correlation changes dramatically. Now, in a scientific study, we can model all of this. We can basically introduce factors, and we can calculate, you know, how much more neutralization against wild type or binding against wild type you now need in order to have protection against Omicron. But this is completely impractical for any clinical purpose. And so I think there's basically only one useful scenario anymore for serology in clinical use, and that's really to check if an immunocompromised patient actually made a response to the vaccine. And if yes, that just means, yeah, they had an immune response. If no, that means they are at very high risk, and they should probably know about that. So that's the only use that I see right now. And before I end, just one slide about therapeutics. As you all know, sotrovimab is not used anymore. This still worked well against Omicron, with BA1, but it doesn't work well against BA2, BA2.12.1, BA4 and 5 anymore. I've shown you data that bebtelovimab still works against all Omicron subvariants. It neutralizes all of them very well. So that's certainly an option. Molnupiravir is around. This is a polymerase inhibitor, which has about a 30% reduction in hospitalization and death, which is not that great if you compare it to Paxlovid. Paxlovid shows an 89% reduction in hospitalization and death, and both of these drugs work well against different variants. So it's not like the variants escape from these drugs. Although we see with Paxlovid that often when it's given and then the course of treatment is over, that the virus rebounds to a certain extent. You might have seen that also. There's no scientific studies on it yet, but I think there's a few coming that describe it. And then for prophylaxis, Evusheld still works against Omicron, although it neutralizes some Omicron subvariants less than others. And with that, I'm just going to give you some outlook. The evolution of SARS-CoV-2 remains unpredictable. We do need updated vaccines. The problem is that right now we didn't have a regulatory pathway to update them quickly, and the FDA is working on that. And we might have updated vaccines by fall. We still need improved SARS-CoV-2 vaccines, including ones that induce mucosal immunity and maybe multivalent ones that give you really broad immune responses. People are also working on universal coronavirus vaccines, but I think we shouldn't fool ourselves. This is going to, if it works, this is going to take many years to develop. And the other important point is, and I didn't talk about that because I'm not a specialist, but in this, but long COVID is real. we don't understand it well. It's often many different symptoms that might have many different underlying mechanisms, but we know that vaccinated individuals with breakthrough infections can get it too. And I don't think we know how to treat it well. So this is really still a problem that's out there. And the other problem that I see is, even if SARS-CoV-2 becomes a seasonal virus and more like a common cold virus, so a nuisance for the general population, it still remains a very high risk for immunocompromised individuals, and we need to keep that in mind, make sure that we protect them well in the future. And with that, I'll end. And unfortunately, I went over time. Sorry about that.
Thank you. I mean, we're so lucky that we have scientists like you working on this. I think it gives us some hope for the future. Florian, I know you had to leave because that's why we made it a half an hour earlier. Do you have time for a couple of questions? If not, it's okay. We'll bring you back again against your will sometime in the fall. But
yeah, I have to drop off, but what I can offer is, if there are questions, if you can put them in an email, I'll try to respond to them later on, if that is acceptable.
Yeah, sure. And thank you so much for taking the time out. I know you made it early just for us, so we appreciate you, and we'll have you back again soon. And thanks to everybody for joining us. Go out there and fight the good fight. Speak to you all later. Thank you.
Thank you.